I'll try to do a little better. Not making any promises though. Yeah, so soil salinity is a major abiotic stress of plants that has dramatic negative effects on agriculture, and is a stress that's only increasing in prevalence due in large part to human activities such as land clearing and irrigation. And so I'm going to talk about some, strikingly, polyploidy has been shown to enhance salt tolerance in plants and a wide taxonomic range of plants, and I'm going to talk today about work using synthetic polyploids of Arabidopsis to try and understand the genetic mechanisms that are underlying that phenotype. I'll start by acknowledging the people who've done this work. Much of the work has been done by undergraduates at Reed College, primarily Cecile Kenny and Susa Oram, but also Eli Sobel, Leo Gartner, and Aidan Turan. Jeff Doyle was the PI on this grant for the NSF that funded some of the work I'm going to talk about today. And some of the synthetic polyploids used in this work were generated by Dana Robinson while she was a PhD student in Adrian Roeder's lab, also at Cornell. Oh, and there was some additional funding from Reed in the form of senior thesis and summer fellowships for these guys. Okay, so as I said, salinity stress is a major abiotic stress of plants. It's estimated that 950 million hectares of land, or 7% of the total land area on Earth, is salt affected. And that's typically defined as having a salt content of 40 millimolar or greater. And this is enough to significantly impair growth of most plants. This includes 75 million hectares of farmland, and then within that includes 20% of all irrigated farmland. And this results in an estimated annual loss of $27 billion. Strikingly, as I said, polyploidy seems to confer salt tolerance. For example, in wheat, this study showed that growth and survival increased, going from diploid to tetraploid to hexaploid, both in synthetic and natural hexaploids of wheat. These are allopolyploids. And then this study of autotetraploid apple found that the tetraploid seedlings had a delay in necrosis and a less severe accumulation of necrosis over time under salt stress. And just to give you a sense of the range of taxa that this polyploid phenotype has been demonstrated in, these are a few examples down here, including a monocot, a number of different dicots, and even a chlorophyte green alga. And this also includes Arabidopsis thaliana. Conveniently, this study by Chow et al. In 2013 showed that tetraploids have prolonged survival under severe salt stress than their diploid progenitors. And this is in 3 different accessions: Columbia, Landsberg, and Warschau. And then under non-lethal salt conditions, they also showed that the polyploids achieve higher seed set. So although under unstressed conditions, polyploids produce fewer seeds than their diploid counterparts, under this 100 millimolar salt, the reduction in seed set is lesser in the polyploids, so that the polyploids actually end up producing more seed than their diploid progenitors. So what are the mechanisms for this polyploid-induced salt tolerance? We're taking a candidate gene approach in which we are starting with diploid mutants of Arabidopsis. So they're mutant for genes known to play roles in salt tolerance. We order knockout mutants from a stock center and then treat them with colchicine to produce synthetic polyploids. We can screen for tetraploids using flow cytometry, where we just identify polyploids based on the absence of a diploid peak in the corresponding flow histogram. And then once we have the polyploids, we simply phenotype the tetraploids and compare them to the diploids to see if this enhanced salt tolerance phenotype persists in this mutant background. And so the general logic of our screen is that we start with this observation that in wild type, the tetraploids have an enhanced salt tolerance relative to the diploid progenitors. So if we plot some measure of salt tolerance, in this case percent germination over time, we see that the tetraploids shown in red have higher tolerance than the diploids shown in blue, 100, say 100 millimolar salt. So then we do the same kind of assay for one of these mutants, a diploid tetraploid pair in a mutant background. And because the mutants are in genes known to be involved in salt tolerance, we expect there to be a decrease in tolerance relative to the wild type. But if within that mutant background the polyploid continues to show an advantage over the diploid, this suggests that that gene that was knocked out must not be essential for that polyploid phenotype. If, in contrast, we no longer see a difference between diploid and polyploid in the mutant background, this suggests that that gene was essential for this enhanced stress tolerance that we observe in the wild types. Okay. So the candidate genes we are starting with are all involved in dealing with excess sodium ions. And plants have three general strategies for dealing with sodium ion toxicity. One is removing those ions from the cell, so as the sodium accumulates in the cytoplasm, the plant pumps it back out with a sodium transporter. Another strategy is to exclude these sodium ions from the photosynthetic tissues. So as the sodium is accumulating in the cytoplasm of the roots, they somehow prevent that from getting into the xylem stream and going up into the photosynthetic tissues. And alternatively, they can just sequester the sodium in the vacuole. So in terms of this first strategy, the key players here are in what's called the SOS pathway, or salt overly sensitive pathway, which consists of three genes, SOS1, 2, and 3. So when high sodium concentration is perceived by the cell, it causes a spike in calcium, which activates SOS3. It then recruits SOS2 to the plasma membrane, where it activates SOS1, which is a sodium proton antiporter. It starts moving that salt out of the cell. In terms of excluding ions from the aerial tissues, HKT1 is a key player. HKT1 stands for high affinity potassium transporter, which in this case is a misnomer because it's actually a sodium transporter. It's a member of a potassium transporter gene family. So what it does is it seems to be involved in basically pumping sodium out of the xylem sap back into the root cells so that they don't go up into the photosynthetic tissue. And then NHX1 is another sodium proton antiporter that's involved in sequestering sodium ions in the vacuole. We have so far successfully generated tetraploids from these four genes and conducted the phenotypic assays, so I'm going to present data on those four today. And we're still in the process of screening culture ploids for NHX1, so we don't have data on that one yet. So our general assay is to grow plants in petri dishes on MS media supplemented with various concentrations of salt, and then simply monitor various performance attributes, germination, growth, and survival over time. We plant both diploids and tetraploids on the same plate, about 30 of each, and then simply quantify these parameters every day or two over a 3-week period to get a sense of the relative salt tolerance between the ploid levels. Okay, so here is our germination data for the wild type diploid and synthetic polyploid. That's sort of our baseline. So we've got percent germination on the y-axis and days post-cold treatment on the x-axis, with the tetraploids in red and the diploids in blue. And we can see that under the highest salt concentration, though there's a delay in germination in the tetraploid relative to the diploid, ultimately the polyploid achieves higher germination efficiency than the diploid does. Okay, so now I'll show equivalent data for the genes in this SOS pathway. So here's SOS3, where we see that the tetraploid maintains essentially 100% germination efficiency, in contrast to the diploid, which shows an impairment in germination efficiency. And we see a similar pattern for SOS2. Okay, so the fact that the polyploid seems to be maintaining an advantage in terms of germination efficiency over the diploid in the absence of these functional copies of these two genes suggests that those two genes are not essential for the polyploid phenotype. Somewhat surprisingly, SOS1, the transporter, the mutants all showed 100% germination efficiency under the two salt conditions that we measured. So this assay is inconclusive. Essentially, we need to test these at a higher salt concentration to determine whether or not the polyploid was doing better than the diploid. Okay. So here's the survival data for those same three lines. So in the wild type, again, we see that the polyploid outperforms the diploid in terms of survival, at least at 100 millimolar. And we see the same general pattern in all three of the SOS mutants. So the polyploid is doing better than the diploid in all three cases. SOS1 is particularly hypersensitive, but you can see that the lower salt concentration of the polyploid is doing better than the diploid, and that's true for all three of those genes. So again, this suggests that none of the SOS pathway in general is not required for this polyploid phenotype. Okay, so now I'll switch to HKT1, which is this transporter involved in pulling sodium out of the xylem sap, preventing it from moving up into the photosynthetic tissues. So in this case, what we're seeing appears to be that the polyploid no longer has an advantage in terms of germination over the diploid. They're both impaired at 100 millimolar salt concentration. And in contrast to the case with SOS1, because they are both impaired, this actually, we think, indicates that there's a loss of this benefit in the polyploid. So that suggests that HKT1 may in fact be critical to this polyploid phenotype. Oh, 2 minutes worth. Okay. Same for survival. The data is not super clean, but whereas it looks like the polyploid may be doing better initially, by day 18, this advantage seems to be lost in terms of survival. Where in contrast to the wild type, where the polyploid advantage seems to be only growing as far out as 22 days. So again, this suggests that HKT1 is critical for this phenotype. So coming back quickly to these conceptual diagrams, we see that the SOS pathway all shows this kind of pattern, which is consistent with the SOS pathway not being critical to the polyploid advantage, whereas HKT1 shows this pattern, suggesting that it is critical for the polyploids to achieve enhanced tolerance. And these data are consistent with some preliminary RNA-seq data we have that show, well, let me just orient you here. We're showing expression level on the y-axis for diploids and tetraploids, either under no salt or 100 millimolar salt for the SOS genes and for HKT1. And so we can see that if SOS1, the SOS2 and SOS3 rather, the diploids actually modestly increase expression under salt stress, whereas the polyploid actually goes down, if anything. So that sort of is consistent with the fact, with the notion that this SOS pathway isn't a key player in the polyploid phenotype. In contrast, HKT1 seems to be constitutively overexpressed in the polyploid relative to the diploid. And then this is also consistent with, again, the fact that HKT1 is known to be involved in minimizing sodium accumulation in photosynthetic tissues. And this study by Chow et al. That I mentioned earlier showed that sodium accumulation is significantly lower in the tetraploid leaves than diploid leaves. And the same pattern is observed in those hexaploid wheats, where they have significantly less sodium in the leaves than diploids and tetraploids. And this actually correlates with higher HKT1 expression in the hexaploids in this study as well. So we think that there are multiple lines of evidence suggesting that HKT1 is a key player in this polyploid phenotype, which is consistent with our genetic screen, which is encouraging. It suggests that this approach we're taking is useful for sort of dissecting the underlying genetic mechanisms. And so going forward, we're going to expand on the screen by, as I mentioned, doing the same thing with NHX1 transporter involved in sequestration in the vacuole. And then we're also going to look at various regulators of HKT1 and other salt signaling pathways. I better stop there, but I did want to just give a quick plug for a special issue of Frontiers of Plant Science I'm hoping to guest edit on artificial polyploidy in plants. Here's the link to the topic. anyone's working with artificial polyploids, I encourage you to take a look and see if you'd be interested in submitting a paper here. Abstracts are due August 31st and full manuscripts. I'll stop there and take questions if there's any time.